Hello, we are Group 4. We'll talk about C3 convertase.
Before discussing C3 convertase, let's discuss complement. Complement is a collection of soluble proteins present in blood and other body fluids. And with antibodies, they support the immune system. In the complement system, reactions occur one after another in rapid succession. Activation of the complement system helps attack pathogens in innate immunity. For example, in opsonization, activated complement binds to pathogens, making it easier for macrophages to eat them. C3 convertase plays a central role in this complement system. There are three pathways of complement activation: classical, lectin, and alternative, and all three pathways activate the C3 protein. Then C3 convertase decomposes C3 into C3A and C3B. So C3 and C3 convertase are very important. C3 convertase activates the complement system and various immune responses. Through this, various immune responses are activated. We will now describe the reactions of complement activation. C1 pathway is initiated by C1 protein that bind directly to pathogen surface or indirectly to antibody bound to pathogens. C1 activates C4 and C2. C1 cleaves the C4-C2 peptide chain, creating C4A-C4B and C2A-C2B. And then C4B-C2A complex is C3 convertase.
C3A and C3B, which result from cleavage of C3, have different functions. C3A is a small peptide that binds to specific receptors and helps induce inflammation. C3B binds to pathogen surface and acts as an opsonin and takes the next step in the complemental pathway. The lectin pathway is initiated by soluble carbohydrate-binding proteins that bind to particular carbohydrate structure on pathogen surfaces. In this reaction, MBL and MASP complex bind to surface. MBL is mannose-binding lectin, and MASP is MBL-associated serine protease. The latter pathway is the same as the classical pathway. The alternative pathway can be initiated by spontaneous hydrolysis and activation of C3, which can bind directly to pathogen surfaces. In other words, this pathway is not mediated by antibodies or sugars. At the beginning, factor D and factor B is activated. Then C3B and BB from complex act as C3 convertase.
To these slides, C3 convertase are C4BC2A and C3BBB. They work differently. C3BBB is stabilized by binding to plasmid protein called properdin. Properdin can also bind to cells and lead to the deposition of C3b on these cells and facilitating their removal by phagocytosis. C3 convertase plays a central role in the complement pathway and assists in pathogen attack. The complement pathway amplifies signals and also allows for regulation of the pathway. Thank you for watching. Have a nice day.